Everyone. Everybody. Welcome to our webinar tonight. One little person's still stuck in the waiting room. I added them in. Yeah, join us. Tell us so you can hear us. Let us know that that you're there, where you're joining us from. That would be wonderful. And from New York, is that Truckee, Carly? Is that how you pronounce that? BC, Utah, Oregon, all over the place. Vermont. All right, awesome, guys. Well, welcome to the webinar tonight. As you can see from our wonderful cover slide, our topic is Epigenetics and Your Dog, and Jessica's presenting for us tonight. For those of you who don't know me, I'm Melissa. I am your host tonight and your tech support tonight, so I will be monitoring the chat box throughout the webinar. If you have any problems hearing us, or if you're having any problems with your connection, you can always type in there and I will do my best to help you problem solve and see if we can't get it fixed. The two biggest things that I like to recommend, and that I will likely be recommending if you have any issues, first and foremost, if you close out anything else on your computer that's using the internet, that'll improve your connection to us. So you're more likely to hear us well and less likely to get glitchy video or any of that. The other thing is if you do get sound issues, and you seem to be the only one, it's probably a connection on your end, unfortunately. So what I usually recommend is you just try refreshing your screen. And sometimes just leaving the webinar and coming back in, that just fixes it back up, and then you can hear us again without any problems. We don't have any videos tonight, because Jessica's awesome like that. So I don't have to worry about any problems there, so everybody should be good on that front. We are, however, recording our webinar. So afterwards, you'll be able to watch it again, if you so desire, and I'm sure you will, because there's going to be lots of good info here. It usually takes us 24 to 48 hours to get it up in your webinar library, and once it's up, we'll shoot you out an email just to let you know it's there. In order to access that recording, you'll need to log into your FDSA account, and then on the right-hand side, just go to Webinar Library, and you should be able to find all the webinars you've purchased there. If you are brand new to FDSA and you have no idea what I'm talking about with your FDSA account, when you signed up for this webinar, you should have also received an email with information on that. You would have received your receipt, you would have received your confirmation email for the webinar, and you would have received a welcome to FDSA email. If you did not receive that email, don't know where it went, or simply don't remember how to get into your account, no worries. You can email me or you can email Terry and we will get you straightened out. My email address is allthethings@fenziacademy.com, and Terry's email address is just help@fenziacademy.com. And either one of us will get you figured out. We'll get you your access to everything fixed up. That is everything on my little house cleaning list, except I'm supposed to open this before, and I forget every single week to talk about what we're doing next week. We'll be back the same time in the same place next Thursday with Mariah, and she'll be talking about reward placement. So her webinar is titled Get Your Cookies Here, and she's talking about how you can use reward placement in your training for better training. And to encourage more animation, more drive, more enthusiasm, and to actually help your dog learn behaviors more quickly. So I hope some of you come back to join us for that. It should be a fantastic topic. It's one that the FDSA instructors got on a whole thread talking about in their private group. So it's definitely an interesting and exciting topic. That said, I'm going to go ahead and hand things over to Jessica. And if you need me, I'm here, but otherwise, it's all you.
Okay, so you broke up for me a little bit there, Melissa, but I am now full screen, so I assume that I should start talking. And can everybody just make the... Because I'm worried it was my connection. Oh, Melissa can hear me. Awesome. Great. All right, and I will get started. So I just wanted to give you guys a quick background on sort of what my perspective is and what my background is so that you know where I'm coming from. I am a veterinarian. I am not in practice. I don't enjoy medicine. I know that's kind of weird, but I really love research. I love figuring out what's going on biologically with animals rather than trying to figure out how to fix them. So I'm a research veterinarian. I have a PhD. I'm in research 100% of the time. I'm particularly interested in dog behavior. I initially thought I would become a behaviorist, but I ended up going into genetics. And so I'm a behavioral genetics researcher, particularly interested in canine personality. So I am not a good person to ask training questions. You can ask. I'm liable to refer you out to the other awesome FDSA instructors for that. But I am a really good biology questions about, well, what's actually going on in the body of the dog when this happens. I'll be talking about epigenetics tonight. So my specialty is genetics and genomics. I have designed epigenetic studies. I went to Siberia and pulled blood from fox kits for an epigenetic study, but we never got them and analyze it to design it so that we could apply for the money, which we then failed to get. So the studies that I'm going to be talking about tonight are not my studies. I'll talk about my research a very little bit, but I will talk about really classic studies in the field, epigenetics, really well understood ones. So that's where all that is coming from. So the story of epigenetics, I think for a lot of people, we feel like starts before what genetics was and how evolution happened. So some of you may have heard of Lamarck, this guy who before Darwin and before we understood evolution, had this hypothesis when people were trying to understand questions about how species changed over time. And so he gave this example of giraffes. He said, how did the giraffe get this crazy, crazy long neck? And the answer that he came up with was, well, maybe there were these animals, precursors to giraffes, and they had shorter necks, and then they would stretch to try to reach the trees, and that would stretch their neck out. And then with a slightly longer neck, then their baby would inherit the longer neck that the parent had stretched, and then the baby as it grew a bit more. And so basically he was hypothesizing that the experience of the parents, where the parents stretched their neck, was passed on to the baby. And of course, we don't these days think that happened at all. Darwin came along and helped us understand a lot better. He worked with finches initially rather than giraffes. But the way he would have explained the giraffe idea is that it's not that the parents stretched their neck, obviously. It's that it was just better to be someone with a slightly longer neck. And so those giraffes happen to have slightly longer necks, just like some humans have slightly longer necks than others. Those giraffes, it was easier for them to get food and therefore easier for them to reproduce, and so they passed on their genes. And he famously looked at this with finch beak shape. So his main contribution, or one of his critical contributions, was that you are born with your genetics. You don't change. Evolution acts on what we call standing variation, differences that are already out there. And that is how species change over time. And it is not about the parent's experience, and the parent doesn't pass on their experience. So these days, we would say that that's really obvious to most of us. But then this epigenetics phenomenon, people started realizing that there are maybe some ways in which your experiences do seem to be passed on to your offspring, and they don't change the offspring's genetic sequence, but it does seem possibly to change how the body uses the genetic sequence. And so some people like to sort of jokingly say, oh, epigenetics, it's like Lamarckism. It's like Lamarck was right. There's some ways in which I think it's a funny thing to say, and there's some ways in which it's right, and I would say there's more ways in which it's wrong, but I'll go into that in detail later. But so if it's true, like what does that mean? If the parent can pass on their experience to their puppies, then does it mean that if a mother dog has particularly good or particularly bad nutrition, that the puppies could be more likely to have metabolic problems? So maybe they'd be more likely to be fat and more likely to develop diabetes. Less of a sports dog problem. I imagine maybe some of you have fat dogs. Absolutely a pet dog problem. So something that's interesting to a lot of pet owners. And then even more of a sport dog problem is this question of whether a mother could learn to be afraid of certain things and then pass that fearfulness on to her puppies. So obviously we really want our competition dogs to be confident. We want them to be able to go in and to work with you. We want them to be able to focus on you and not focus on that scary dog who's around the corner, not be overwhelmed by the new environment, but be able to be confident and solid behaviorally. So this is a really particularly interesting question to some of us, and for me, trying to understand what the biology is underlying canine personality and trying to piece out how much of that is environment and how much of that is genetics. And now there's this epigenetics piece to fit in as well. So a very interesting question for a lot of us. So it is certainly true that it makes sense for parents to pass on hints to their kids about their environment. I found this picture that I'll use repeatedly throughout this talk to illustrate that. So in the picture, it almost looks like it's a very intentional thing where the mother is thinking about what to pass on. And of course, it's not like that in real life. Of course, in real life, it's much more that the mother's body is the one making choices and they're not conscious choices. But mom is passing on all kinds of stuff to her offspring. She's passing on information about what the environment is like that the baby's going to be born into. It's really, really useful for babies early on in their development to have some information about what their world is going to be like. As their body starts to develop, it's going to have to make choices. It's going to have to go down certain pathways developmentally and not down others. And so it's going to, go down some pathways and some decisions about how to develop its metabolism versus the stress system. And it's good for it to know early on what kind of a world it's going to live in. And so moms absolutely pass that information on. I'm very interested in hormones personally, so the examples that I give would tend to be that mom passes on that information through hormones through the placenta. The placenta is a very, very active organ that passes on a lot of information about how stressful the environment is. And so the placenta itself almost is making some decisions. Mom passes on information about that for milk. And she certainly passes on information from her own behavior that the puppy can learn from. So yeah, it's useful for the puppy then to learn from that and to make some of these decisions. Is the puppy going to have a really safe life, plenty of food? This is the life that our pet dogs are more likely to have, or is the puppy going to have a life where they need to be afraid about what's coming around the corner and they're going to need to worry about where their next dinner is going to come from? And it's not pathological for a dog to develop to be obsessed with food, to be hypervigilant and very worried about strangers. It's just appropriate in this particular street environment. It's not appropriate for a pet dog environment, and something that we definitely want to avoid. So the way that the dog saves a lot of this information, there's a variety of ways that the dog is going to save this information that the mother and to some extent the father passes on. But one of the ways that this information is saved is through epigenetics and markers on the DNA. And so here you can see this strand of DNA, which obviously this is a tiny fragment. It would be much, much longer. You can see the double helix. These are each would represent letters, A, C, T, or G. And sort of fancifully, they have these markers here glowing. They obviously wouldn't glow in real life, but so that we can see them very clearly. These markers are placed on the DNA. The sequence isn't changed, but these markers are placed there as a way of storing this information and helping the body know that the way the DNA is used should be changed. There we go. So then one of the questions that you folks let me know on Facebook that is very interesting to you is this concept of transgenerational inheritance. It's the concept that parents are going to pass on this information to babies through epigenetics, and that it's going to be stored. The parent is going to store some information and pass it on much, much later, and then it's going to be passed on for multiple generations. So there's definitely information passed on in the uterus, in the nest, in milk, and I'll talk about all of that in detail. But there's also this really open question, this very, very interesting question about whether a parent can store some information and much later in life pass it on to their offspring. And the media, the popular media, has really, really hyped studies that suggest that this does happen. So just a quick spoiler alert as I'm going through this, and I'll talk about the transgenerational inheritance stuff in detail. But what we do understand pretty well at this point is that very general information does get passed on to kids, information about how much food to expect the environment to have and how stressful to expect the environment to be. So that kind of general information is mostly passed on after conception. It's mostly not held on to and then from before conception and passed on later. This very specific stuff There's a really intriguing study that I will talk about, suggesting that perhaps very specific fears could be passed on. That kind of stuff. There's some interesting stuff out there that I'll talk about. Don't really have, in a deep way, understand exactly what's going on or how it works. There's a lot of people studying about it, and it's absolutely getting reported in the media, but it's something that I hope you will all take a bit of a grain of salt when you read it. So this concept of what I call transgenerational inheritance, this idea of passing information on to future generations. I had trouble finding a good picture for this, so I went with a AKC pedigree to show you the concept of future generations. And so the idea there is that your puppy, and genetics is obviously works this way, right? Your puppy gets some genetics from his grandfather. His father may not have shown, may not have expressed some of those traits, but he got them from his grandfather, and it's, there was no experience involved, just got passed on over many generations. But so then there's this question of thinking back to Lamarckism, is it possible for a parent to have an experience and pass that on over multiple generations to their offspring? And it turns out to be kind of hard to tell about this, but there is a classic study, of course, in humans, the Dutch Hunger Winter Study, which some of you may have heard of, and I have a picture of Dutch food stamps here. So this happened during there was a famine in the Netherlands during 1944 and 1945, towards the end of World War II. There was a German blockade, which cut off all food and fuel shipments from farm towns into places, to cities in the Netherlands. And it was a winter of unbelievable famine. Around 18,000 people in the Netherlands died of malnutrition that year. Adult rations at one point were cut to less than 580 calories a day by the end of it. And so really tough time for people. And what was really interesting is that children who were in utero during that famine grew up to have an unusual set of diseases. They were much more susceptible to things like diabetes, obesity, cardiovascular disease, and other health problems such as schizophrenia. And if you think about it, in some sense, in some way it makes sense. Their bodies learned very, very early on, while they were in utero, that the world was a really difficult place. It was a place where there was not enough food, and it was pretty scary. And then, however, things changed, right? They had this short experience at a very formative time. Their body made some conclusions from, and presumed that the world would be that way always. The world, thank God, wasn't that way always. But as a result, their bodies had made some decisions and then were having difficulty adapting to the way the world actually was. So you can imagine that a body that makes some metabolic decisions, presuming there's really not going to be enough food, then when it is presented later on with an excess of Twinkies, has real trouble managing that. So that was a classic study, and one in which people first started looking at, can experiences be passed on? But it turns out that, in fact, the babies were there during the famine. It wasn't so much that the mothers had experiences during the famine and passed them on later. It was that the babies were in utero during the famine, and so in a sense, the babies were actually there. So it actually was the babies having their own experience. And there's this interesting concept of, if you have a mother bearing a female fetus, there's a way in which 3 generations are actually all together there. And there's this lovely, a lot of images about this on the internet. There's this lovely one here. See the mother, who's the first generation, and she has her female fetus, who's the second generation. And the female fetus, while in utero, is already making the reproductive cells, the eggs. So females make their eggs while they're in the uterus, and those are the eggs that they're going to have for the rest of their lives. So you have 3 generations together experiencing that famine. So those babies were there during the famine, and depending on how you really want to define and look at transgenerational inheritance, I'd say that this is actually not really. It's that they're there, and you'd have to look at the next generation to say that this is not that it's not an experience that the baby is having. And so it's a little different with males. So males make their sperm throughout their life, and I looked up for humans. A sperm has a lifespan of about 77 days. In a human, I didn't look up what it was for dogs. I'm gonna guess it's something fairly similar. So over the course of 77 days, then you could say that the baby, or half of the baby, is innocent there. So for this 77 days, less than 9 months, but there's still this way in which that part of that puppy is there already experiencing what's going on. And so to think about something as transgenerational, I would argue that it has to be passing on experience that was acquired before conception if you're female, or before the sperm was made if you're male, arguably before the eggs were made if you're female. And that's really hard to test for, right? So if you test just sort of a normal average human family or dog family, then there's a lot of questions you have to ask. You have to ask, did mom or dad's behavior affect the baby? So I was poking around trying to find some studies for you guys for transgenerational inheritance, and I found one where they injected the moms, I think they were guinea pigs, with stress hormones, and then they saw changes in the baby's behavior for several generations. But they didn't foster the babies out. So first of all, they didn't do IVF. So first of all, the babies were growing up, developing in the uterus of these moms who were presumably somewhat stressed out. So the uterus was passing them information, and then they were raised by these moms who were somewhat stressed out. And the babies, that presumably is going to be very likely to change the baby's behavior. And so that's the first generation, and then these babies had their own babies. Well, they already presumably have this opportunity to inherit learning from their moms and their dads about how stressful the world is, and they can easily pass that on just as this normal way of sort of learning from each other, and so not necessarily epigenetics. You also want to ask, is the baby raised in the same environment as mom and dad? So again, I said, epigenetics is really about the body learning, mostly at a young age, about what the environment is going to be like, and then recording it in the DNA. And so if the baby is raised in the same environment, so think about these street dogs, right? You have a street dog. It maybe learns at a young age that it needs to worry about food and worry about scary things, and it can record that in its DNA. It has a puppy. Now, is it passing that on epigenetically, or is the puppy in the same environment and learning exactly the same thing? So if you control for both of those things, where you ideally artificially inseminate and then possibly, ideally, even take the baby out of the uterus and have it actually grow up in the uterus of another mom and be raised by another mom and be raised in an entirely different environment than the biological mom and dad. And then remember as well that there's all these babies there's 3 generations together with females, so you want to look multiple generations out. So these are the kinds of studies, these are the kinds of things that people have to do in these studies to really look at true transgenerational inheritance. So it is really difficult to do. And the epigenetic mechanisms that we understand that I'm going to talk about in this talk don't actually seem to do this. There do seem to be some studies suggesting that some of this information can be passed on. Again, very general information, not mostly things like how much food there's likely to be and how much stress there's likely to be. There have been studies showing that these things can be passed on multiple generations, and very well controlled studies in the ways that I just described. But the actual epigenetic mechanisms that we do understand, we know get reset generation to generation. So, however, I feel like that thing that epigenetics do each generation of helping the baby save the information about their environment and be prepared for the proper environment is an incredibly cool thing and very much worth understanding. So I'm going to start out talking about that, and then I'm going to go on to talk as well at the end about some of these studies that really seem to be showing that some of this transgenerational stuff does happen, but that we sort of don't really know exactly how it's happening. We're presuming that it's epigenetic, but we don't know that it is. So to summarize that, right, so during pregnancy, there's some really important information being passed along. And I always wonder then about the effects of stress and malnutrition and the effects that it'll have on puppies. None of this stuff is black and white, right? Lots of great puppies come from moms who are stressed out. There's plenty of stuff interacting together to make a dog's personality. It's not just how stressed the mom was when the puppy was in utero. It's the actual genetics that are passed on, and it's how the puppy was socialized and how the puppy was managed. So all of that interacts together. However, I personally think it's very important to keep pregnant dogs out of shelters, get them into foster care as soon as possible. A lot of shelters are liable to have pregnant dogs stay in the shelter until they give birth, and then they understand that the puppies need to be raised in a home, but I feel that the pregnant dogs need to be in homes as well. I also know, more relevant to the sports community, that there is sometimes a practice of purchasing a pregnant dog and shipping her overseas. Doesn't happen all that often, but it does happen, and that also sort of gives me pause. So some stuff to think about. There aren't studies looking at this, so I don't have direct evidence that this actually affects puppies. It's just that the information that we have suggests that it might, and so it's something that would be something I would think about carefully. All right, so how does epigenetics work? So obviously, baby starts out as a single cell. Egg and sperm come together, make a cell, and that particular cell contains a complete set of DNA. And DNA has instructions for what a cell should do, no matter what cell type it is. So your complete set of DNA has information for all cell types and all life stages. So when this baby is a single cell, there are specific genes in the DNA that are useful for that. There are genes that have that information about how to divide and grow. There's genes that have information about how to form organs, how to make skin and legs and fur. And then there's other genes that will be useful not when you're only 2 or 4 or 8 cells. There's other genes that will be useful later on. Genes that have information about how to think, how to make decisions, how to eat and sleep and have sex. All of that information has to be on hold while you are a tiny little 2-cell stage, but it has to be able to be useful later on. So as the cell divides and goes through many stages, then it has to keep track. It has to know these are the genes that I need to use right now, and those are the genes that I have to use later. And so it has to remember, where am I? Am I at the 8-cell stage? What genes are useful for me now? Or now, am I a liver cell? Am I a kidney cell? Am I a brain cell? So what genes do I use if I am any of those different things? And so this is the basic and most important and probably least interesting to us use of epigenetics, is saving information for a particular cell about what genes it needs to use to operate. Really basic stuff about what type cell it is. And so you can think of this as bookmarks, pointing the cell to the relevant genes, the relevant chapters in the DNA. This is the stuff you should be using now. It's relevant to the cell type that you are. It's relevant to the life stage that you're at. You can learn this other stuff. So that is the basic thing that epigenetics does, and without it, we would not be able to be multicellular organisms. So those genes then that are bookmarked, they're going to differ, as I said, based on cell type, but also based on environment. And so it's not just information about being a brain cell, it's also information about, well, if you're a brain cell in a very calm and safe environment, these are the genes that will be useful to you. But if you're a brain cell in a super stressful environment, then these are the genes that will be interesting. So, I have here some pictures of the different environments that dogs might find themselves in. So that then, as well, is what epigenetics ends up being useful for, more subtle use of it, to say, okay, well, it seems that you have determined that this is the kind of environment that you're going to live in. You're going to be a competition dog, a show dog, a military working dog, a pet, a street dog, and so different genes will be useful to you in those different environments, and it's useful for the body to have bookmarks to all of those things. So those bookmarks are epigenetic markers, and the study of those bookmarks is epigenetics, and they do affect your DNA, but they don't change the sequence, and they change how the DNA is. So how is that different from genetics then? How does that work? So some specific dog examples. I'm going to start out with non-epigenetic examples so that I can make sure you have a solid grounding as we go into the details of epigenetics. So people want to start like really with details, so I'm giving it to you. So this FOXI3 gene is the gene that makes a dog be hairless or not hairless. So if the dog has, so everyone has 2 versions of each gene, right? You have mom version and dad version. So if a dog gets 2 versions of the gene that's normal and that works normally, it makes hair and teeth. If you have 2 versions, you're going to be a normally haired dog, as most of the dogs in the world are. If you have 1 working version and 1 broken version, you're going to be a hairless dog. And so there's a lot of different hairless breeds that have many of them have this particular mutation. I believe not all of them. And as it happens with this particular mutation, if you have 2 broken versions, you're non-viable, meaning you're not going to make it out of pregnancy as an embryo. So that is how FOXI3 works. And to demonstrate how this works. By the way, this lovely graduate student in the lab where I work was kind enough to draw some of these pictures for me, and would, I'm sure, love feedback on what makes sense and what doesn't make sense. So here we have this strip of DNA, and she has illustrated normal DNA. It starts out, what turns green is that bit where there's a gene. So chunks of DNA are genes and chunks that are not. Talk about that more in a minute. So we have this bit of green here, which is for the gene, and we have basically a little robot. It's an enzyme called RNA polymerase. It's a little robot floating around, comes down, finds the DNA, lands on the DNA. So here it is, landed on the DNA, and it basically opens up that DNA and makes a bunch of copies of it as RNA, and those copies get transformed into protein. So this would be the normal way that this thing works. RNA polymerase comes down, DNA, makes a whole bunch of copies, a whole bunch of normal genes. Now, if you have a mutation in the DNA, here's mutation, then you have this little robot coming down. He's trying to read off the code about how to make the gene. You have the mutation right there, and the mutation is made in these RNA copies, and you have broken non-functional proteins. Proteins don't work, and you have a dog that has some trouble making hair. So that's the traditional genetics that we were taught, basically in high school, right? For a lot of us or college, that genes code for proteins. Proteins either work or they don't work. If they don't work, then you get these different traits. And that is very much how we thought about genetics until fairly recently, really in the last couple of decades, that it was really all about proteins that either worked or didn't work, and that's what's. But we're starting to realize recently that regulation is actually much more important, particularly for some of the really interesting traits like behavioral traits. So another fantastic Kathleen image here. This MITF, MIDF gene has to do with how much white spotting a dog has. And so you can have a dog with a lot of brown and not much white. You can have a dog with more white or a dog with a whole lot of white. And it ends up being the case that this has to do not with breaking MIDF, but with how much of it is made. So it's an entirely functional protein, but we make either more or less of it, more brown or more white. And so what happens is this gene is used early on during development. And there's some cells that migrate around the body, and if the cells successfully migrates all the way around the dog, then you have a fully or mostly brown dog. If they don't get quite so far, the bits that they don't get down to stay white, and if they don't get very far, then most of the dog is white. And the reason for that, so we have a bit of skin here with some fur growing out of it, and we have these cells down here. These cells are melanocytes, and they are what is basically injecting color into the fur. So if the melanocytes make it there, then you're going to be brown, and if they don't make, or some other color, obviously there's other genes that modify that, if they don't make it there, so this indicates here that something is broken, then there's no melanocyte there, it can't inject color, and so you have some white. So in other words, this then has to do with, during development, a difference in how much is made of this gene that causes the melanocytes to move. Not as much of the gene is made, the melanocytes don't move as far. Does that make sense to you guys before I go on, this idea of regulatory differences and how far the melanocytes move? Yeah? One person, so we're good. Okay, I'll go on. So, so regulatory variations. So, again, I think something that was thought quite a few decades ago was that most of our DNA contains genes that code for proteins, but it turns out a tiny, tiny part of our DNA does that. Only 1.5% in humans, and again, probably very similar in dogs. And so we don't know exactly how much of the rest is for regulation, but a lot of it is. And so we have this massive chromosome, right? And so the scale is changing here as it comes out. We have this little bit that's a gene on the DNA, and then you imagine that a whole lot of much of the rest of it is actually used for other things, and we're just starting to figure out what some of those things are. So I talked before about this idea that there was this enzyme called RNA polymerase, which you can think of as like a flying robot, floating around, and it lands on the DNA in order to express the gene. And so you can think of it as like an airplane landing on a runway. And so these regulatory mutations can make it harder or easier for a gene to be expressed by changing the runway. And you can think of it as like a permanent change to the runway because this is genetics. So it's, this runway is too short, and so it's hard for planes to land, and so not many planes land there. Some planes will still land there. It's still possible. It's just not all that easy to do. And so if you imagine that there's a lot of these little robots floating around, and they're trying to land, but it's just that much harder, then maybe not as many are going to land there. Maybe they're going to go find a better runway. They're going to go land someplace else and express some other gene, but not this particular gene. And so that is how we think of regulatory mutations. Here's the robot, he's trying to land, and here's our little mutation, which we haven't read, and so it's trying to land, but there's something wrong. That mutation makes it not work so well, so not quite so many of them land. But then once it gets there, it successfully makes the healthy proteins, right? So you're still getting these proteins coming out, fully functional, working just the way they're supposed to work, but just not quite so many of them. We'll compare them here, how we have perhaps a dog who has a lot of brown is successfully, has a good runway, no mutation there, successfully having this robot land, successfully making a whole lot of these little proteins which are going to make the melanocytes move during development so that they get all the way down there and you don't have so much white. But then you have a dog that has genes coding for more white. He has this little mutation there. It's harder for the plane to land, and see you get this difference in number. And so again, this crucial point is it's a difference in number. Regulatory mutations are about how much of the gene is expressed. They are not about whether it works or not. It works fine. So when you compare a regulatory mutation to what I was talking about initially with, for example, that mutation that controlled hairlessness, hairlessness is pretty much an on or off thing. The way that particular mutation works, it's not very fine-grained. Regulatory mutations, the body likes those because it provides very fine-grained control, which is why you can see them in behavioral traits where there's this whole range of what can be expressed. So more fine-grained control with regulatory mutations, but they're still genetic. They're hard to change, right? How much white you have is set when you're in utero. So maybe you have this amount of white set in utero and then you're born into a very snowy world, and it would be really nice if you had more white. You're screwed. There's nothing you can do about it. This is a genetic code, and there's nothing that can be changed after you're born. So the nice thing about epigenetics, specifically that the body likes to use for traits that it wants to be able to tune early in development, is that the control is just as fine as in these regulatory mutations, but it's a lot easier to change at important developmental points. So I'm going to give first an example of epigenetics that is a little bit easier to see because it has to do with mouse coat color. And this is a very, very artificial construct. It's not going to be directly relevant to dogs. But it is a good way to first understand epigenetics, and then I'll go into stuff that's a bit more relevant to dogs. So for sure, these coat colors, these different coat colors that you're seeing in the mice in this picture, there are mice out there that have these coat colors different due to genetics. That gene could be broken in specific ways due to genetics. However, these particular mice, with this very artificial construct, researchers basically took brown mice. This is the natural color for these mice, is brown. They broke the gene, the agouti gene or ASAP gene that makes the brown, the nice, this only brown color, and then they have this mouse with this broken gene. But then they used epigenetics basically to fix the gene. They used epigenetics to tell the body. They had basically inserted some nonsense code into the gene, which made it work. That would work. And they used epigenetics to tell the body, just ignore that broken part, and so then it started working again. So they have this really fascinating system where, based on epigenetics, you can have this fully functional form of the agouti or ASAP gene, and you can be brown, or you can have this fully broken form. But again, because epigenetics are all about regulation, they can have everything in between. And the way that they were able to control epigenetics were used to turn the gene back on, basically, was they basically fed the moms, again, this is highly artificial, they fed the mother mice this diet that was really, really rich in the particular substance that mice use, or that all use, to put epigenetics markers in place. So again, kind of a weird way of going about it, but what they wanted to prove was that this was how epigenetics worked, and that they were able to control it. So going back to that regulatory mutation, this is exactly what we saw before with MIDF, right? Plane tries to land and it can't because there's a mutation, which makes it a little bit harder. And therefore we have fewer copies of the protein being made. So with epigenetics, it's almost the same, except for that we don't have a genetic mutation. We have this marker set sort of on the outside of the DNA here, and preventing the plane from landing pretty much in the same results. You can see in this example, I just had it look like exactly the same result. The difference being that you can't change this after birth, but you can change this after birth. So again, this is this idea of the runway being used more or less. And so rather than thinking about it, the genetic differences with the runway, we said it's something that can't be changed. It's like how long the runway is. Epigenetics, I like to think of it as putting rocks on the runway. Very easy to change, right? And so the plane comes down, tries to land, sees there's rocks, doesn't try, but easy enough to take the rocks away. And that is what then results in these mice looking so different. And so the marker that I'm talking about right here is known as methylation, and that's one of the most famous markers, although not the only one, used in epigenetics. You have this piece of DNA here, and we have methyl groups here put onto the DNA, and again, sort of silly, they have them fall. And so that right there is a methyl group. And so here's the DNA, methyl group is stuck on there, and that is what makes RNA polymerase have so much difficulty. So I just want to give a plug for gene expression studies. So gene expression then is what we call that difference in how much of a protein is made. How much of the gene is expressed and then transformed into a protein. Genetics, as I just said, can affect gene expression, and epigenetics can affect gene expression. And this is what I did during my PhD, is I looked at gene expression in the stress systems of tame versus aggressive Russian foxes. And I used that to find how their brains were altering the genes differently. That was a very clearly genetic system. They've cross-fostered those foxes. They know that what I was looking at, the differences that I was seeing, were definitely genetic. There are a lot of people who look at differences in, well, if you looked at differences in dogs, for example, where the genetics are not so different as they are in these hyper selected foxes, you would know that a lot of the differences that you were looking at were due to epigenetics. Just looking at gene expression, you don't know whether it's genetics or it's epigenetics that did that. But it's still interesting sometimes to just say, I don't care. I just want to know what is the actual difference. I don't necessarily care what we're looking at. So just different things that you can look at. So coming back then for our story about moms passing information on to babies using epigenetics, we're much more interested, I think, in stress. Information about how stressful the world is going to be passed on, much more interesting than this artificial construct of coat color. And so, quick overview of the stress system, which is what I study, what I love so much, is, I think probably most of you are familiar with this concept of stress. It releases this hormone called cortisol. We'll just pretend that this is a molecule of cortisol here. This green thing is floating through the blood, and we have a cell membrane here, some receptors sitting here, and cortisol comes in and it connects to the receptor, and that is how the cell knows that cortisol is out there in the blood. Getting down here to sort of nitty-gritty, which may be a bit much for some of you, is that cortisol then it connects to that receptor, comes inside the cell, comes inside the nucleus, and the receptor is actually going to go attach to DNA and pass information about changing gene expression. And this is a totally normal way for gene expression to work. I mean, we've just been talking about ways in which it can be changed very long-term by genetics, but it actually changes quite rapidly short-term in response to environmental changes, such as the release of cortisol. And so this is one of the ways that happens. So, whoa, sorry, I thought I had put my markers someplace else. So, so then, how does epigenetics come into this? One of the things that happens is that the hippocampus, a part of your brain, has basically the off switch for the stress system, which I love talking about this off switch because I think it's a very, very important part of the stress system. And so when cortisol comes and attaches to the receptors in the brain, basically the number of receptors that are there is very important for how the off switch operates. And so if there are more receptors there, it's a little bit backwards from what you might expect. There's more receptors there, then the brain is very, very sensitive to how much cortisol is out in the bloodstream, and it will turn that off switch off much, much faster. If there's not as many receptors, then it takes longer for the brain sort of to notice that the cortisol is there because it doesn't have as many ears to listen with, I guess is one way of thinking about it. And so if you have fewer receptors, your off switch is not going to be as easy to trigger. You're going to stay spread out longer. You're going to tend to have a stressier personality. So do you guys see where I'm going with this, right? With the number of receptors and talking about gene expression. So epigenetic markers are going to affect the amount of that protein that is the receptor for that stress hormone, how much of that is made in the hippocampus specifically. This is this famous series of studies from Michael Meany and others. He's a famous Canadian researcher who beautifully described this system of how epigenetics interacts with the stress system. And I talked about this a little bit in my socialization webinar earlier, but now I'm going to go into details specifically about how the epigenetics works. And so, as I said in the socialization webinar, there's this interaction where you have these female rats who have different maternal care for their babies. And so we have a rat here doing what we call arch-back nursing. So she's standing up with her back arched, allowing the babies to nurse. And she's also going to do a lot of licking and grooming of her babies. And what happens when she does that is that she's causing changes in the methylation of that gene for that receptor for cortisol, the glucocorticoid receptor, for that receptor in the hippocampus, specifically in the hippocampus. And so how the mother treats the babies very, very early on in their lives, in the first 2 weeks of their lives in mice or rats, is going to change the methylation, the epigenetics around this particular gene, which codes for the receptor that listens for cortisol. It's going to change it only in the hippocampus, not in the rest of the body. And it's therefore going to change how responsive the hippocampus is to cortisol coming in and saying, okay, there's plenty of cortisol in the body now. Time to stop making quite so much of it. Time to start relaxing. You're stressed out. Quite enough. Thank you very much. Time to relax a bit. So that is how that works, and I think it is a really beautiful example and very, very well described and well understood of something that is, I believe, very relevant to dogs. We haven't described the system in dogs, mostly because no one is willing to cut puppies' brains open and take a look to see whether there's methylation in the campus. But I strongly suspect that something very similar to this is going on in dogs, that early on when they're in the nest with their mothers, they're having some interactions with their mothers that may be similar to the amount of licking and grooming that these rats do, or it may be something different for that species. I don't know. But I believe that there are going to be epigenetic changes, probably in their hippocampus, probably over exactly the same receptor, to change and tell them, Mom is telling me that this is a very stressful environment or a not very stressful environment, and that is going to affect how they are going, what their personalities are going to turn out to be as adults. And these these rats, they were tested at the amount the mom licked and groomed them, correlated with how much methylation there was over that gene, correlated with how many of those receptors they produced, and correlated with what kinds of personalities they had as adults. And then they passed it on to their children, although that was easily broken by cross-fostering them. So if you took a rat who had a mother who licked and groomed them a lot, she would lick and groom her babies a lot. And but if you gave her to be raised by a mom who didn't lick and groom her babies a lot, then she wouldn't lick and groom her babies a lot. And so they were passing on that information that they had learned from their mothers based on their own experiences and not based on genetics, but based on genetics. And so then the really obvious next question is can we change it? We know what those markers are. We know that they're super important. And yes, Mimi's lab did do this follow-up study. They did take a look at, there's a particular enzyme that goes and either adds or removes these methylation groups onto DNA. And they were able to, I forget exactly how they did it, whether they broke that enzyme. I think they actually injected a lot of it into the hippocampus. And yes, they were able to change the amount that these animals methylated, and therefore they were actually able to change their adult personality. Super cool. However, totally not ready for prime time. You really don't want someone injecting a test substance that has not FDA approval into your puppy's hippocampus. I don't know how many of the rats had side effects from this. I imagine it was not side effect free. You definitely you would really like to be able to target that specific area of the DNA, that specific gene to be changed, and we have no idea how to do that at this point. You just changed the whole DNA. At least they were able, through the injection, to target just that area of the brain. So you could imagine in a dog, well, should I get a shot, right, IV? But it would go to the whole body, and it would change methylation of the whole body. And while this Mini system is that, like Mini described, the stress system is super well understood, it is one system, and there are a zillion other systems that we do not at all understand. And I personally would really love to learn more about them. I think this is a direction I would love my own research to go at some point, although I'm still hung up on this problem of not being able to look at the puppy brains. So then another question that we might ask is, well, if an animal, has epigenetic methylation set early on in life, is it able to change over time, or is it set early on and then that's it forever? And so the answer there is apparently it does. Did you ever have this lovely picture of Nancy's dog Reason earlier in her life and later in life? But the study I'm going to talk about was done with identical twins in humans. And so they looked at them throughout their lives, and they saw that, yeah, there are changes in methylation throughout life. It does seem to change in some places, not in other places, and it does so quite gradually. So we don't really know exactly what areas are going to change and what areas aren't. So we sort of looked at it in some humans. We don't know. Is the body perhaps using this as a way of keeping track either of environmental changes or the fact that it's aging? We don't know. Something that was super interesting that was found in this, however, was that there's some difference between identical twins and non-identical twins, in that identical twins are genetically exactly the same, and their methylation seemed to change in very similar ways. So if you looked at them throughout life, they might differ somewhat from where they were earlier in life, but they would be more similar to each other, though, at the current time. But if you look at people who have slightly different genetics, you see that they change, their methylation changed in slightly different ways, which suggests that there is some sort of interaction between epigenetics and genetics. And so if we're thinking about, where we're going to put those methyl groups in the DNA, it's possible that a certain variant of a gene, an certain variant of the gene might be a really good place to put a methylation mark for whatever reason that we don't understand, or it might be a really bad place. And so it might be that certain versions of genes are more likely to be methylated than other versions. And there was a really lovely series of studies done. This one gene, FKBP5, it's a gene that is associated with the stress hormone cortisol. Again, for those of you who understand this level of biology, it is a chaperone protein, and so it chaperones the glucocorticoid receptor while it folds, and so it can help the glucocorticoid receptor fold in different ways and therefore be more or less sensitive to cortisol. So, and this particular gene, it turns out, can be methylated, so its epigenetics depends on different genetic versions. So you look at somebody who has a particular genetic version of this gene, and they are more likely to have more methylation on it compared to someone who has a different genetic version of the gene. So what does that mean? It's this thing called gene by environment interaction. And oh gosh, guys, I feel like I need to warn you here at this point before I dive into this, but I seem to be 4 minutes before 10 o'clock, and I'm on slide 52 of 70. Okay, so I'm just going to apologize ahead of time for those of you that I keep up late, although I know that a lot of people expect there to be 2 hours for this, and I know there's a lot of questions. But so sorry, I have to spoiling. So gene by environment interaction is how genetics and experience interact. So in this case, I found this lovely example here. So on the y-axis here, we have body size. So down here, you're small, and up here, you're big. And then on the x-axis, so here, we have whether this particular individual was living in a warm climate, and here we have whether he was living in a cold climate. These are the two interesting ones. I'm going to leave them for last and go through the basic ones first. So these two dots are going to represent the different climates, and the two different colors you see here, but black versus red, are going to represent different alleles or different genetic backgrounds. So here's a bunch of different possibilities that you could have. You could have that it's exactly the same no matter what. You have exactly the same body size no matter what these two different genes, the two different versions of the gene look like, and it also doesn't matter what environment. You can have that if you have the red version of the gene, you're going to be smaller, and if you have the black version of the gene, you're going to be larger, but that doesn't matter what environment you live in. You can have that if you live in a warm environment, you're smaller, and if you live in a cold environment, you're going to be bigger, and it doesn't matter what version of the gene you have. Or you can have, if you live in a warm environment, you're going to be smaller. If you live in a cold environment, you're going to be bigger. And if you have the black version of the gene, you're going to be a little bit bigger than if you have the red version of the gene. But basically, it's the same interaction with the environment. Now, where it gets really interesting and where it comes back to this FKBP5 gene that I've been talking about is when the slope of the lines is different. So if you're going to draw the lines out and they cross at some point, that's when the slope is different, and that's when you have this gene by environment interaction. So what's going on here, that you're going to be the same size no matter what version of the gene you have if you're in a warm environment. But if you're in a cold environment, then all of a sudden the black version of the gene is going to make you bigger. Red version doesn't do anything. So that's interesting, right? It's that in order to be bigger, you both have to have the black version of the gene and you have to be in the cold environment. Here we have them doing opposite. So if you're in a warm environment and you have the red version of the gene, you'll be little. And in this case, the black version makes you bigger. But suddenly, if you go into the cold environment, you see exactly the opposite. So again, and this is the kind of stuff that is so hard for geneticists to unravel when you have these interactions. So it's like, wait, it's the opposite of what I would think it would be. All right, so what does this actually mean for personality? And they did this study, obviously, in humans. They looked at children, and they looked at, super difficult study to do, by the way. They had to find people who had died and donated their brains to science in order to look at the methylation levels in the hippocampus. The hippocampus turns out to be this fascinating part of the brain for PTSD and stress. So first we have, so we'll just say that FKBP5 has a normal allele versus a risk allele. So the normal allele, if the person has a normal allele, they have no childhood trauma, then they're less likely to develop PTSD as an adult. If they have a normal allele and they do have childhood trauma, they're still not going to develop PTSD as an adult, and we would call that a resilient personality. If they have the risk allele and they have no trauma, they're okay, they're still not going to develop PTSD. But if they have the risk allele and they have the childhood trauma, and they are at high likelihood of developing PTSD, and it happens that we also see epigenetic changes, they actually have less methylation in that particular situation. So that is a gene by environment interaction. Okay. So what we're talking about then is how epigenetic markers get set at particular, very, very important points during development. And so particularly that very early stage in puppies, which is going to correspond to that first 2 weeks in mice. It's a bit longer, more like 5 to 7 weeks in puppies. And also, almost certainly, although it hasn't been studied, but I would imagine when the puppy's a bit older, I think there's still probably some of this stuff going on when the puppy first comes home to its permanent home and is going through that socialization process. So we know that this stuff is set at particular important points during development, and that it probably changes a little bit later on with age, but it's difficult to change later on. But we don't understand it. This is the output of a study that looked at the association of methylation with chronological age. And so what they're looking at here is a bunch of different chromosomes in a human. So this is chromosome 1, chromosome 2, chromosome 3, all the way up. And this is the level of significance. And so if these dots rise above the level of significance, then what they're saying is at this position, in this chromosome, there's something going on here. There's more methylation, which seems to be associated with chronological age. What we don't know is what that means, right? Well, what's its correlation. So what's cause, what's effect, what's the mechanism, what does it mean? Is there something that we might want to change? Sometimes methylation seems to make gene expression be increased rather than decreased. Why does that happen? And I've been focusing on methylation so far because it's very, very well understood. The other very well understood mechanism is acetylation. So right here you're looking at these. This is a long strand of DNA, this blue. And DNA is not just generally entirely loose in the cell, so it gets tangled. And so we wrap it around these proteins here. So this is how DNA at a high level tends to live, as these strands that are wrapped around these proteins. Acetyl groups are attached to the proteins rather than to the DNA. And you can see how this part of the DNA back here is very, very tightly wound up. That makes it really hard for that airplane to land and express genes in that area. But up here, the DNA is much looser. So you can see how easily the plane can land here and express the genes in here. Much harder for it to do right here. And these acetyl groups have a lot to do with that. So those are other really important markers that are used. And again, we still don't really understand how they get set, where they get set, really hard to tell. So one solution that I wanted to mention is just to ignore the markers, but to look at DNA accessibility, or to look at gene expression, or to look at both, which I think is what is being done in this particular study that I'm about to talk about. This is a study that's still underway. It hasn't been published yet, but it's coming out of my lab, being run by a fabulous researcher, Catherine Ward, a super smart person doing this really interesting work, looking at differences in basically in how open that DNA is, as we just talked about, in cells in the saliva in dog and wolf puppies during that early socialization period. And so it seemed likely in this case, obviously, that there could be some genetic differences between dogs and wolves. I think once she understands what's going on there a little bit more, obviously the differences between dogs and wolves would be much more dramatic than the differences within dogs. But I can imagine that once we're able to map this out a little bit, it might then be very interesting next to go take a look at the differences between different puppies or different breeds of puppy during the socialization period. So again, genetics can be one of the things that affects how open and closed that DNA is, but epigenetics also can. It certainly can have to do with experiences that these puppies had in utero. And so this is more and more, I think, an approach that researchers are taking is just to look at these differences either in gene expression or in what we call chromatin confusion, which is the accessibility of the DNA, which is in the DNA. So look at that rather than look at the epigenetic markers. Because frankly, we just don't understand the epigenetic markers. We don't really know what they mean. Sometimes we look at those things together and try to figure it out from there. More and more, I think genomics is going in the direction of looking at a lot of different things all at once and trying to put them together, which I think is a great direction for going. But that's sort of where we stand right now. The other thing I want to mention, and I think I've mentioned this a couple of times during this talk, is that doing either epigenetics or gene expression or chromatin configuration DNA accessibility studies, super difficult because you have to look at a particular tissue. And so with behavior, you're interested in the brain. And so I've talked a lot about the hippocampus, and that is a region in the brain, and it is changing a lot during that early socialization period with puppies. And I would love to be able to be in the Star Trek world and take a Star Trek scanner and just scan a puppy and have it tell me, you know, it has this many receptors for cortisol in the hippocampus, and this is how the DNA is methylated, and be able to look at how that changes and relate it to the maternal care that the pup is receiving. I think we'd be able to learn so much if we could do that. But all we can do right now, well, we could kill the puppy and take the brain out, and no one wants to do that, and I certainly don't want to do that. Another thing that we can do is what was going on in this study here, what Catherine Ward is doing here is taking saliva. And other people, particularly with humans, sometimes will take blood. We like blood because it goes around your body so that you can take it out without killing the individual, but also goes up into your head and goes into your brain. So we have some idea that hopefully these differences that we see in the blood may actually correlate to differences that are going on in the brain. But we don't know that for sure. And so, you know, for example, when you're trying to get funding and you say things, and this is in large part why we didn't get funding for that Fox study I talked about, because you'd say, oh, I'd really like to be able to take blood from these kids during their socialization period and look how it's different in the tame and the aggressive foxes, the methylation in the blood. And they come back and say, well, but it's not the brain. How do you know it's going to be the same in the brain as in the blood? And the answer is, I don't know. I just hope that you'll give me the money because I don't want to kill the fox kids. So that's very much where epigenetic is why you see a lot of these studies being done in rats and mice. I'm not willing to do those studies, so I work in dogs doing the best that I can. We are currently collaborating with a veterinary school. It's a very long, slow process, but I'm trying to get them to get us brains that are donated so people can bring their dogs in to euthanize them in the end of life. Sometimes those people are willing to donate their dogs' bodies to the vet school for science, and the vet school has agreed in some cases to let us have some of those brains. We got two recently so far, and I actually, it's still Thursday, right? I earlier today actually had lunch with the person that I'm collaborating with on. We're talking about all the awesome things that we're going to do with those brains. They're not puppy brains though, but it's a start. So so back to transgenerational inheritance, which I'm sorry to be so down on, but you guys are really very interested in it, and definitely do want to talk about it. And it turns out, so the first course I ever took on epigenetics, one of the first things she said is, I still remember this moment of listening to this lecture, and she was like, and so epigenetic markers are stripped when the eggs and sperm are made, and then they're stripped again at conception. And I was like, what? How can it be? But how is this stuff passed on? It is stripped, and if you think about it, it kind of has to be stripped, right? Because I talked about how part of what epigenetics does is it tells you about what's going on, what's useful for you to be if you're a particular kind of a cell. And it's cell, there's really no way, there's no mechanism that we understand for you to save the information about, if I were a hippocampus cell in a stressful environment, how would my epigenetics look? Well, you have to strip it because instead you're one cell and you have to save the information saying, this is what I do next. So I divide into two cells so I can make a baby. And so all of this stuff is stripped. The methylation, the acetylation, the configuration of the components, and the DNA accessibility is all stripped. Except for maybe not all of it. And so there have been some studies that have found that there are some regions that aren't stripped. And we super don't understand which regions are not stripped, why they're not stripped, how the information is passed on, all of that kind of information. That pass. So I did want to emphasize that me say that I talked about, which I think is just the most relevant one to dogs and dog behavior, not transgenerational. That information was passed on in the early couple of weeks that the baby spent with their mom. And Mimi knows this because he actually went and took a look at brains of the babies when they were first born, that they were born without methylation. And then depending on how much mom licked and groomed, the methylation built up, and he was able to chronicle how the methylation built up. So we know that that's definitely due to early life experience and not transgenerational. Some of these studies have shown some of this information being passed on, particularly metabolism, widely studied in humans because we care about obesity in humans a whole lot. Response to toxins, so a lot of these environmental toxin studies, particularly looking at like BPA and things like that, also looking at sniffing and secondhand smoke. This information, some of this does seem to be passed along transgenerationally, and so some of these studies are not so well controlled, but some of them are, and some of them have really successfully, I think, demonstrated that at least the epigenetic mechanisms that, at least the way that we understand the information might be passed on is all controlled for, and yet the information still does appear to be passed on. We don't know how that's happening, right? We know how the epigenetics works, and we suspect that there's got to be some epigenetics on passing this information on. We don't know exactly how. So we have this super interesting study, and I went forth about talking about it because we don't understand what it means all that well. And Denise specifically told me that she's in it, and I was like, well, she's paying me, so I guess I should talk about it. This one came out in 2014, and I remember I was in my PhD program when this hit the press, and I got emails from people about it being like, have you seen this? What's going on? It's crazy. So they looked at fathers, which are a little bit easier to look at than mothers, because remember, mothers, you have to look at more generations, but fathers. So they, what the researchers did was they paired the smell of cherry blossoms which is a smell that they didn't expect the mice to have experienced in any other way, and they didn't expect there to be any sort of genetic pre-programming having the mice have one feeling or another about it. So they paired the smell of cherry blossoms with foot shock in these male mice. And then they tested the offspring, and they were very careful. I think they even artificially inseminated those. So you could imagine that there was something where like the males became very, very stressed out because of this. They reported that they thought it was mild foot shock, but I remember thinking at the time, like, well, how do you know what's mild to a mouse, right? So they could have been very stressed out and passed that information on to their mates when they mated with them, but they artificially inseminated, so the fathers had no interaction with the mothers. And then they tested the offspring, and they did it. It was really interesting test called pre-pulse inhibition. So it was not that the mice smelled the cherry blossom smell and became frightened. That wasn't it. What they did do was, if you play a sudden mouse, that's when the mouse is going to start. If you have the mouse smell this cherry blossom smell, and then you play a loud sound, the mouse startles more than if it smells a different smell. So there was this very specific fear that appeared to be passed on, and that appeared to be passed on for 2 generations as well, so not just 1 generation. So would you call that transgenerational inheritance? What do you guys think? And while you people are typing, I'll answer Brianna's because it's right there. The answer is I don't know if they took the sperm from the fathers before the peer conditioning as well as after. That would be a really nice control. I don't know if they did that. I'd have to go look at the study. I did want to say that I'm going to be on Facebook after this. I might actually go to bed, but I'll start a Facebook thread and I'll answer questions that are going to require me to look things up. I'm happy to look things up, but if you ask again, Brianna, on Facebook, if you are on Facebook, that will remind me to look that up. Carly and Piper think this is transgenerational inheritance, and I think it is too. It's a really spooky study, though. They controlled this really well, and I did some reading because I was like, I got to talk about this with them, and they're really going to want to hear about it. But so here's the thing about it: it hasn't been replicated again. Other people haven't done it, and so that always sort of gives you pause. And I was sort of hoping, like, it's okay. Well, it's been 4 years now, so maybe someone's replicated it, but apparently no one has. I should find a blog post from a blogger who I respect, I think he was Neuroskeptic, who said, their results are a little bit too clean, they make me a little uncomfortable, and then the researchers answered that, and they said, well, but there were other less clean results, and we just put that in the appendix, basically, in the supplemental materials. We didn't put it up front for everybody to look at, but there's some shady results, we put those in the back, they're there for everybody to look at, and so there's sort of not replicated, meaning no one else has published a paper saying that they have tried to do it and succeeded, or that they have tried to do it and failed to do it. It's possible that people have tried and, for whatever reason, not published the paper. And I don't know why that would be, but there's no more information out there that I can find about whether this works. And I did hunt around. And it's possible now that one of you would be like, oh, you missed this thing, and that would be great to know. But I looked and I haven't seen it, and I tried to find anyone sort of weighing in on like, well, what does this mean? It's super specific, right? Like it pairs a cherry blossom smell with a foot shock, and so these offspring now think that even in the presence of cherry blossom smell, we're going to be a little stressier. So I just don't even know how the receptor for the cherry blossom smell would be a gene, and in fact, these researchers did look and see methylation differences over that gene, but associating that smell with something bad, that's not something that would go into DNA so far as we know. That's something that's encoded in your neurons. The researchers did look and see differences in the neural pathways of these mice, but we don't know how that would be passed on. So it's spooky. It seems to be a good study. It was published in a good journal. It seems to be very well controlled. They seem to have been very responsive. So I don't know. Sometimes we don't know. So was Lamarck right after all, right? Is it possible that parents pass on their experiences to their offspring? And you guys can answer that however you want. I would say that epigenetics is usually only stable for 1 to 2 generations. I would say it's definitely not over evolutionary time. And so I wouldn't use the term Lamarckism, which really is about evolution, particularly because really what epigenetics is about is passing on information from parents to offspring and saying that this is what the environment is going to be like. Try to adapt yourself to it. But it's really important then that if your grandmother said something and then your mother might say something different because the environment's going to change. That's the whole point. It's supposed to be something dynamic, and so it really is something that shouldn't be passed on over multiple generations. I mean, if you were to pass on the information to your offspring that the cherry blossom smells scary, and then your offspring never experiences anything associated with cherry blossom smell, why is it worth it for them to pass it on to the next generation, right? Like, they should be passing on the information that the environment has changed, and what was true for grandpa was not true for me, and so you shouldn't do it. You shouldn't make assumptions based on that. So getting into my summary then, as I start to wrap up, so we learned about how genetics and epigenetics and environment all interact together, and I think we really sometimes like to read some of the stuff out in the media or learn about these cool things and think that it's going to be super powerful on how our dog's personality is designed and how the personality develops and grows. And genetics is going to be important for your dog's personality. Epigenetics and those early experiences are going to be super important. The current environment and how you manage your dog is going to be super important. All of those things are important. But I do think that it's useful to think that very early on there's some information that's your dog's brain and other body parts, but the brain is what we're really interested in here, that your dog's brain is receiving some information early on that's going to affect how it develops. And so that doesn't mean that the dog can't change. And I have my shy dog, who I've talked about before, who's changed dramatically over the now 7 years that I've had her, dramatically but slowly. And so I would say, yeah, probably she did have some epigenetic markers early on, which programmed her to be a certain way. She absolutely can be changed. I'm really glad I didn't give up on her, but it was slower because she had those marks programmed early on in her life. So epigenetic markers can be difficult to change, but it's possible. And the study where we look at the change in humans over time shows that they do change. We don't really understand what makes them change. It's probably going to be different for different ones, for different traits, but they're definitely changeable. It's just difficult. True transgenerational inheritance, that really spooky cool stuff, probably does happen. Some studies suggest that it happens. I think it happens a lot less often than the media suggests. And just when I was doing some reading for prep for this webinar, I found a couple of cases where I talked about the guinea pig one, where the mothers were injected with a stress hormone, but then were allowed to raise the babies themselves. Where you sort of have to really take it with a grain of salt and think about how well the study was controlled. Some of them are very well controlled, some of them are not. The media is typically really bad at differentiating between those, and it's a much better headline if you can talk about this spooky inheritance of experience. Okay, so quick plug, I'm going to be talking about this stuff in my Biology of Building a Great Performance Dog class in June. I'll talk about epigenetics there. More than happy to talk to you there about your individual dogs and answer some of your questions about the dogs there, although obviously I'm happy to do it here as well, and thread on Facebook. On the alumni list, which you should all have access to. So I'll start that right after this, after we are done with our questions here, and you guys can continue to ask questions, and I'll answer them for as long as you have them. Probably not tonight. I'm probably going to go to bed after this because I am on Eastern time. But I'll start the thread before I go to bed, and then I will look for it when I wake up in the morning, and hopefully you guys will.
This one,
This is my puppy, Dash. He's a year and a half old now, but here he was at 9 weeks. It was like the second day I had him, back when he was not being a jerk yet, and eating through his $250 hobbles, which are there to preserve his shoulders after his $8,000 surgery. She did this morning. She's why I'm so stressed out today. But very quickly, he was in the middle of medicating his pain right there. Okay, so hit me with your questions. I'm gonna drink some water.
Yeah, lots of. Go for it. So I've got a bunch of questions here. Guys, if I did not reply to you and tell you that I had your question, I may have missed it, so feel free to retype it. Also, I will do my best to take in your questions and agree to them as I ask them. But sometimes it takes me a second just to get back to you, just because I am talking and reading and talking things. All right. So the first question that came in was from Brianna, And she was curious if there are, if you know of any connections that have been seen between allergies in a mother affecting the puppies or allergies in a father affecting the puppies.
I don't know. Fortunately, this is going to fall into the cognitive behavior. So if you ask me about behavior stuff, I'll be like, oh yeah, I saw this study. I don't know. I have not seen any studies about allergies being passed on. It seems hypothetically it could be a reasonable thing to pass on, but I haven't seen any studies about
that. I asked if you have a dog who started out a little fearful, a little sensitive to some things, but you work with them and increase their confidence. Does that work that you do make the dog to make the dog confident help future offspring?
Good question. Yeah, I think so. So we have this really well understood system for the stress response with moms for sure. So let me talk about moms first. So for sure, if that dog were female, she's going to pass on a lot of information about what her perception of the world is to her puppies. And if her perception is that it's a terrifying world, we have my dog Jenny. I don't know how much of her issues are genetic and how much of them are due to her crappy early socialization. I think some of both. But even if I were confident about her genetics, I would never breed Jenny because I'm absolutely sure that she would pass on information to her puppies that the world is terrifying. Because to her, the world is terrifying. So absolutely, if you have a confident dog, they are going to tell their puppies in whatever way, and that is going to be through hormones in the uterus, that is going to be through hormones in their milk, that is going to be the level of maternal care that they give puppies, and there are just starting to be some really interesting studies coming out about level of maternal care that dogs give their puppies, and we still don't really know what we're looking at or for or what the effects mean, but people are starting to look at that, which I'm so excited by. So I think that's going to be something really important to look at. And so absolutely, a mother dog is going to pass all of that on. I mean, even at the level of, a person comes to see the puppy and the mother dog freaks out and barks or cowers back. Puppies are going to learn just from that, but I think there's all kinds of other ways that a mother dog is going to pass that information on. For a father dog, it's a bit less clear. So ways that a father dog can pass that information on. There is some evidence, not studied in dogs, mostly studied in laboratory rodents, that if a father is very stressed out, that when he has intercourse with a female, he will treat her differently, probably be rougher, and that she will pass different information to her offspring based on that treatment. I haven't read up on that system very carefully, so I'm not going to be able to answer in more detail about that, but certainly the way he treats the mom can affect the offspring, at least in laboratory rodents. I don't know if that's true in dogs, but it would be possible. There's some interesting studies recently showing that dad passes on some information, and I debated whether to talk about this and decided I was already going into enough detail. But dad passes on some information by passing on some messenger RNA in sperm. So we're just starting to look into that and to understand that. So basically then what happens is that at the single cell stage, the embryo comes pre-primed with information from dad saying, start out making more of these genes. RNA does not last very long, like on the scale of hours. It degrades very, very quickly. And so I wouldn't call this epigenetics, but it is another way that the father passes information. So it's going to affect the embryo at the single cell stage. And there's some studies where they're starting to see like that seems to affect them as an adult, or how or why. So that's some really interesting stuff. So to answer your question, yes, I think that making anyone that you're thinking about reading should absolutely be as confident as possible.
Excellent. Sharon, I'll let you know that I have your question. I will ask it. I will warn you, though, that it's the end of a very long list of questions, so you may have to wait a little while for the answer. All right. Carly noted that you partially answered this already, but she said if I could ask it anyway, that way if you have anything to add, that would be great.
Sure.
So Carly and Piper, are more things only passed matrilineally? For example, the mother has fears passed during pregnancy and whelping versus a male who obviously only contributes DNA in most cases.
Yeah, I mean, I I think that is pretty much what we're seeing, is that more information comes from the mother. But I think it's also one of those things where scientists make assumptions early on, and so obviously the mother must pass more information on, right? The mother is the female and is mothering and nurturing, and what does the father have to do with the babies? The father is out being the breadwinner. So there's all these sorts of cultural ideas that we bring to our research, and so I think that early on, a lot of the research was looking at the maternal effects. And in fact, so I'm this too, right? So I'm very interested in what I tend to refer to as maternal effects, but we should really call them parental effects, because they can come either from the mother or from the father. So I think people started out studying maternal effects more. People are starting to realize in the last decade or so that maternal effects actually can be a lot more interesting than we thought, and there's a lot more going on that. And so I think there's more studies out there showing maternal effects than showing paternal effects.
So interesting.
And so I would hesitate to say that reflects ground truth. That it is very possible the father has a 50% genetic interest in the success of the offspring, just like the mother has, and so he has plenty of incentive to pass information on to the offspring. And I think just because we focused on the mom doesn't mean that he's not doing that. On the other hand, mom has all these extra opportunities to do it, right? So I think the answer is I don't know. I think that there are more studies about moms.
Excellent. Brianna asked, any research done on connections between the microbiome and genetics?
Yes, I am sure there have been. So I think what you're saying is it so just as I talked about how the different genetics that you have can affect your epigenetics, I think you're asking is, does the genetics that you have, can that affect your microbiome? I am sure that it can, and I'm sure there have been studies done on it. I am not up on my microbiome studies, however. It's a bit out of the scope of this webinar, so I can't give you a more detailed answer than that. But I just I'm confident that it must be the case.
She said and vice versa in the comments for what it's worth. So
yeah, I can't think of a way that that your microbiome would affect your genetics. I think it could only go the one way, that what genetics you have would probably make your gut a better or worse environment for certain bacteria, and so it would affect your microbiome, or even your habit. Like, I like to eat chocolate. That might affect what bacteria I have. But I can't see how the bacteria that you have would affect your genetics.
So another question, and I think this was while you were talking about the hairless dog stuff. Brianna said, any case studies of mutations such as FOXI3, I don't know if it's how you're pronouncing that, in mosaic expressed females.
Brianna, can you type out?
I can copy
she's really active. She's typing it. Yeah.
That was the original question.
Oh, FOXI3, yeah. Are there any cases of mutations such as FOXI3 in mosaic expressed females, the hairless breed mutation? So you're basically asking, is it that, no, mosaic expressed females meaning like X inactivation, or meaning animals that are true mosaics, because it's not on the X. I don't think it's on the X. I don't actually know what chromosome it's on. I just assume it's not on the X.
For 1 minute, it's like 11 o'clock.
You're not typing, Brianna. Oh, there it is. Yes, you are. Okay, so yeah, I don't think it's on the X. So I think what you're thinking of is X inactivation, which is, for those of you who don't know, it's basically how you get, for example, calico cats. Is that the genes that affect whether the cat is has like the black or the brown or whatnot is on the X, and because females have 2 X's, and that is twice as many X's as you really need, as males have demonstrated. In every cell, one of our X chromosomes is sort of tied up and not functional, and it's the other one that's used. And so if you have different genes, we talk about you get one gene from mom, one from dad, so you have one chromosome from mom, one chromosome from dad, and so then sort of early in development, and we decide which chromosome is going to be tied up of the X chromosome in females, and so that's why you get these splotches in calico cats. By the way, I did neuter a male calico one time. Super cool. I walked around the shelter being like, look what I just did. Males can be calicos if they're XXY, 2 X's. But I don't know how that would work in hairless, because I don't believe it is on the X, and so I don't believe that would happen.
She had one more question that was related, so I'm going to just paste it in there. Is the MITF gene related to Irish spotting also?
Yes, I believe it is. We're actually, I have not generally been interested in coat color, but we're doing this project with my lab with the Darwin's Dog website where we're trying to, starting to finally have the money to sequence some dogs. And so we're trying to give people information back about like, well, your dog's genetics predicts that you will be blah, blah, blah. And I think it is known. I think MITF is a large part of what controls white spotting, but there's like different ways that MITF is regulated, and so there's like a bunch of different mutations around it. And I think there's like 6 or something, and so like they can combine in different ways. And I think it's that one particular combination is likely to give you that Irish pattern, which is what my dog has. For those of you who don't know what Irish pattern is, you have a picture of it right there. Oh God, I just don't remember though, and it just makes my head hurt. I'm like, why do I have to learn about coat color when I don't learn about behavior? Apparently,
Interesting. All right, so Carly had 2 questions, and I think they're related, so I'm gonna ask them together.
Okay.
So, and they're on coat stuff again, so a little more on that. But the first question was, wow, so with the hairless gene, does that mean that hairless dogs have a quarter-haired dog, half hairless, and a quarter that die in utero?
Yes. Yeah. So, and so I was actually double checking that because I knew someone was going to ask me that question. And so again, there's a couple different breeds that can be hairless, and apparently there's some different mutations. And so I'm talking about this one specific mutation that it is in the Mexican hairless, which is at least the breed that I'm the most familiar with. And so yeah, it's sort of like with Merle, where you don't really want to have a double Merle, so you don't really want to have a double hairless. But you have to in order to have a hairless at all. And so yeah, so you definitely probably really don't want to breed 2 hairless dogs to each other. You kind of want to breed a hairless dog to a haired dog. And apparently this is recognized that in the Mexican hairless, some of them will have hair. I didn't realize until I did this reading for this webinar. And so some proportion of the puppies will have hair and some proportion will be hairless. But if you breed 2 hairless together, then some proportion of them will be homozygous hairless, and that means that they will not make it out, so you will see smaller litter sizes because some of them will die in the
So the other question Carly had was, so why are colored hairs often reliable patterns? She said that's where she was getting lost when you were talking about that stuff.
Yeah, right. So I didn't get into that. I'm not sure that is known, and I definitely don't know it. So what I was talking about, it's very hard to give good, I like to give good real life examples. When I first started teaching genetics to dog people, I was like, well, I'll just use the original Mendelian examples because those are great. So he studied beans or peas, and so it's like this could be a yellow seed or this could be a green seed. Super boring, but at least it was like very, very clear what was happening. But dogs, there's just fewer genes that are really easy to understand. So the white spotting is clearly controlled by more than just that one MIDF gene. MIDF gene, by the way, a lot of those mutations in the MIDF gene were discovered by my boss, Eleanor Carlson. And so when I was having trouble understanding, I would read some of the papers. This was not for this webinar, this is for Darwin's Dogs. I would go down the hall and be like, hey, Eleanor, what does this paper mean? And she would tell me. So that was really nice. But it is spotting, but not all of it. And so there's clearly some other genes interacting to give us these predictable patterns. And I don't know if some of that is understood or not. I kind of suspect it hasn't actually been discovered yet. And then there's some amount of randomness at birth, right? So you know that if you clone a dog that has white spotting, the clone is probably not going to have exactly the same pattern, and that's because there's just some amount of chance to how those melanocyte cells migrate when you're in utero. But there's also some genetics, obviously. It's more than just MITF, there's some interaction. But it is just talking about just that one gene I thought was a good example to get my point across.
Interesting. So you were talking about the mice with the different colors, that image. And Paula asked, did they feed the moms methylation, MSM? She was curious.
Oh, yeah, I saw that. Oh, it's funny because I was like, oh, damn it, I should go look up and see exactly what it is that they fed the moms. And then I was like, no one's actually going to ask me that. But no, you guys did. I did. It does say it in the paper, and if you ask me later, I will go look it up. It does say. I mean, it's basically like, methyl is a substance, and it's just these little methyl groups that's used by the DNA, but there is a substance that contains a whole lot of it. and that's what they fed the mice, and I just don't remember exactly what it was.
She also asked, she said she'd really like to understand how feeding methyl food sources affect epigenetic triggering. She said she's read different things, but nothing really definitive, and just kind of wanted me to see if you.
Oh, like in real life, as opposed to in the mice. I don't know how it does in real life. I don't do nutrition, so I haven't read those studies. I suspect there is an effect. The thing about this mouse study is that they made it, and I kept saying how artificial it is, because they took this gene and then they looked at really extreme variations. And I don't remember how much they varied the mom's diets, but I bet you they were pretty extreme and much more than you would see in any one of our pet dogs. So I've got to think that the mom's diet does affect the babies. I expect that in natural systems that the mom's diet is mostly going to affect the baby's metabolism. That's what diet is relevant to. I expect it shouldn't affect the coat color very much at all, but I don't actually know what all things it affects because that's nutrition and that's boring. Actually boring, but I have to limit myself to some things I can't know. So,
she did ask as kind of a follow-up in the comments there, if we feed MSM, does that do good or bad things for future generations?
Don't know. MSM, which we don't know.
She was also curious about, so she's seen many references to biblical writings regarding how the sins of the father, etc., gone for multiple generations. So what she wants to know is how many generations changes usually.
Yeah, so I think that's one of the things people are still trying to answer. And I argue in this webinar that mostly it's just going to be one generation. That mostly it should be, epigenetics really, as we understand it, is about the experiences that you had that you are saving in your DNA, and you could be an egg cell or a sperm cell, and definitely you could be an embryo or a fetus for sure. But that in that case, we sort of look at it as one generation because you and your mom both experienced that thing, and that's the really usual case. How long did it go on? Studies, I think I saw at least one study that followed it out. 4 generations studies don't really like to follow it out farther than that. I would argue that at that point you start being really surprised more, because again, it's really supposed to be about recording your experiences, and so really your body mostly should be discounting what happened to your parent or your grandparent. Really your body should be taking most information from what is happening to you. But I think that's very much still up in the air. There's still a lot that we don't understand.
Brianna asked on cortisol, if those receptors affect the animals so much, why don't the behaviors associated with stress fluctuate throughout the day with their cortisol levels?
Okay, so Brianna's mentioning, she's referring to the fact that cortisol levels change throughout the day. So they tend to be, in humans, they tend to be higher in the morning and in the evening. Just a side note, which is kind of funny, I had to do this reading for my master's. Apparently not necessarily true in dogs. There's some studies show that dogs are just like humans, and some studies show that their cortisol levels don't fluctuate quite so much. And I think the conclusion that I finally came to was that dogs who live with people have their cortisol levels act like people, probably because their sleep cycles are similar to ours. Anyways, so that was kind of interesting. So why doesn't their behavior change throughout the day? There's a lot of different ways I could answer that, so I'm just thinking about how the best way to do it is. So one answer is that cortisol is not just about behavior. Stress is not just about behavior. So cortisol definitely is about things like, I guess one way in which it's about behavior is that at night when your cortisol levels are low, you're very quiet and you sleep, and in the morning when your cortisol levels are high, we think that they spike right around the time that you wake up because your body is readying itself for the day. Your cortisol levels also have a lot to do with whether you've eaten or not and how hungry you are. So not seeing an animal get more or less stressed out in response to the cortisol levels being naturally high or low, I am not sure I can give a solid mechanistic answer as to why that is. I think I would just say that Cortisol does a lot of different things in the body. We don't fully understand all the ways that things interact. And when we think about how it interacts with muscle switch, particularly in the hippocampus, which is the best way that we understand how it affects behavior, I'm sure it affects behavior other ways as well, but that's one that we understand. That's really about the number of receptors that are made that are sensitive to cortisol levels, and those don't change throughout the day. Anyways, really the answer is I don't know, but that's the best I can do for you.
Fair enough. Annie and Paul, I'll get your questions at the end here. Got them. All right, so the next one, Nancy asked when you were talking about kind of all that stuff, is that also how SSRIs also work artificially on the stress system?
Oh, I think I saw that floating through. What was I talking about exactly? You said all that.
I think it was while you were talking about cortisol and the number of receptors.
Oh, yeah. SSRIs definitely affect that system, and we don't fully understand how SSRIs work. Should I follow up?
Yeah, if you looked up in the comments, she said one of her questions.
Oh, this is Nancy. Sorry, I missed it. It was Nancy. More of my question had to do with cortisol receptors, wondering if it was similar to serotonin receptors. Can we affect serotonin receptors via epigenetics too? Oh, I haven't seen any studies on epigenetics with serotonin receptors. That would not surprise me at all if that were the case. But I think either no one's looked at it or somehow I failed to see it. I think the latter would be surprising. So I think if there were a study on the epigenetics of serotonin receptors,
and thank you for keeping him on the pills and making that huge effort. I so appreciate it. Lots of exclamation
That'd be cool
points.
to do.
Chrissy was looking for the links to some of the studies, but I'll share that with you later.
Oh, no. I meant to say that to people, and I'm really sorry I waited because some people will start listening by now. But just like last time, I will definitely send the links to you, Melissa, and you can put them on the notes associated with the recording. And so help me remember, but I wrote them up ahead of time this time, and then I was like, I should send them to her right now, and then I didn't. Actively. But we definitely should get those up right away, so that because last time I was worried that people came to look for the recording and didn't see them was there. But I will link to you and all those things.
Mm-hmm. I'll ask if we know how long that initial methylation period is in humans, since in rats I think you mentioned it was 2 weeks.
Yeah, we don't know. I'm really curious about the human socialization period. I'm sure we have one. I think it's much longer than in dogs. We don't know, because it's just super hard to get children's brains in the cats.
She's overtired.
I'd imagine. Amy said...
We probably know more about the... Sorry, I know it's hard because we're off by time, so I can't tell you. But I bet you there's people who know more about the human socialization period than I do, and I bet I just don't read those studies as closely because they're not dogs. So there may be some, but I've never seen an explanation of how long, or even a reference to human socialization period, which is hilarious because we clearly have them. If you raise a child in a closet, it's going to be screwed up. But humans are in general socialized fairly well. So.
Amy said humans shift at puberty in the.
So Amy, you would say that that's the end of the human socialization period is when puberty starts? She started to type. She's probably still typing in my. It's just not showing. She says, I think that's too strong a statement. Yeah, I think that probably. Amy probably has more information on this than I have.
So she did ask, why is methylation the mechanism of interest and not chromatization? Or don't they both affect gene expression?
I am not sure what she means by chromatization.
Let me share her comment. He says no pronunciation is a problem.
Yeah, she may be talking about, say chromatization. She may be talking, it's Amy, so she probably is, yeah. Are you talking about DNA structure, chromatin configuration, maybe?
She's typing.
Yeah. I can see it, and then there's the awkward pause. Yes, and what regulates turning genes on off. Yeah, so absolutely, chromatin configuration, or I talked about that. I think you may have said this earlier, and then later on in the talk, I did talk a little bit about what I referred to as DNA accessibility, which is the same thing as chromatin configuration. Chromatin configuration is, so first of all, I started out talking about methylation because it's so well understood in some of these specific studies that give very good examples, and so I wanted people to understand it. And it's really the first epigenetic mechanism that we discovered, and it's still a very well, it's not a very well understood one. These are specific systems that it's used in are well understood, but overall it's not that well understood. So methylation and acetylation are both specific markers that can last over long periods of time. One of the ways that those markers work is through affecting chromatin configuration and therefore gene expression. And so there's sort of two separate ways of looking at the same thing. It's really, right, unschooling, it's really the chromatin configuration that's the, particularly with acetylation, the important thing. Methylation sometimes affects chromatin configuration. We don't fully understand why. We understand a lot better acetylation. Methylation, though, often also has this effect when it's on the gene promoter that I talked about, where the RNA polymerase is trying to get at methylation on the promoter, and it can't because the methyl loop is there. And we understand that a bit better, and then there's some way in which it affects chromatin configuration as well. I don't believe it's understood yet, although maybe I'm missing certain studies on that. So it all interacts together. And again, I thought about going through this as I was making a talk, and I was like, damn, it's already too long, so let's do this next.
She said great down there. And Carly says the fascinating what you geeks believe.
You're welcome. Yeah. Staying in the questions when the other PhDs come in and ask really detailed questions, and I'm like, screw it, I'm not explaining to anybody else, I'll understand. But, yeah.
Lucy asked, can a marker eventually be removed with exposure to a different environment?
Yeah, so, I mean, I covered this somewhat, and the answer really is probably, but we don't know. So they certainly didn't, it would be very interesting to those mice with Meany's system that he discovered, where, they methylated up the system that we understand really well based on their maternal care, but, now you go and place it in some, really safe, lovely environment, it's an enrichment, and it feels very happy. And then you check later on to see if that changed, if that particular area that we understand changed. I bet you it does over the course of a lifespan. Of course, mice don't live very long. Medias rats, sorry, rats and mice are different. Rats don't live very long. So with humans then who live long enough that we'd be able to see these changes, and we're unfortunately not able to do these controlled experiments, and so we really just don't know exactly what's going on. I believe, from the evidence that I discussed earlier in this webinar, that methylation probably does change over time. We know that there is a particular enzyme or a particular little robot that comes and adds them or removes them. So, for example, cells divide, right? And so you open up the cells to open up its DNA and copy the DNA, and when it copies the DNA, it has to copy the methylation as well. And that's an obvious point where the cell could choose to not copy the methylation if it didn't want to. So I think there are times during life when things could change. We just don't understand when or how or why those things happen. I suspect they do change, but very slowly over time.
Brianna asked, how does epigenetics/gene expression work in instinctual behaviors like herding, and how do those instincts seem to fade in mice that are no longer being bred for those behaviors?
Oh my god, I have no idea. I have no idea. The instinctual behaviors, my gut tells me that that's genetic and not epigenetic, but I don't know. There was one really interesting study a while back, not herding, but they were looking at scent detection dogs. And they discovered that if they let the litter of puppies watch the mom do scent detection work, that the puppies learn scent detection work faster just by watching the mom. So it's very possible that puppies are learning some sort of these behaviors that we think of instinctual from the mom early on. But, with herding behavior, it's so clearly highly genetic that I gotta think that there's not that much epigenetics affected. But I don't know, no one's looked at that. I want to look at that, not the epigenetics of it, but the genetics of it. I'll tell you guys when I get around to that. I'm hoping that that's going to happen in the next few months, but it's dependent on some stuff at work. But I'm trying really hard to get there.
She says new study, let's do it.
Yeah, let's do it.
Carly and Piper, wanted to know, can we ask to donate our dog's brain to science? When we have an old dog, can we set that up sort of like a pregnant mom? Can be set up to donate cord blood for birth?
I would love that. So where we're standing right now, that is absolutely the plan for down the road. Where we are right now is that I'm working with this one vet school, and we're sort of doing this proof of concept thing. They don't want people to come in to do the donations because they're concerned they don't want to turn into the place that people come to euthanize their animals. I think that we could do this sensitively, and I think that there's plenty of people who would be interested in doing this. And I think it also would be helpful right now, the dogs that I'm getting donated, I don't have any behavioral information on what I can do. There is interesting stuff I can do, but there'd be much more interesting stuff I can do if I had behavioral information. But you have to know I'm not going to go to someone who just had to euthanize their dog and ask them to fill out a 150 question survey on their dog's behavior, right? There's no way I can ask that. But if it was people like you, where you sort of planned ahead and we could like enroll you in Darwin's dogs and you fill out all the dog's behavioral information and then we maybe win it. So that is definitely up or down the road, but it's a very long, slow process. It's a very sensitive thing, obviously. The vet school really doesn't want to do it wrong, have people get angry at them. We don't want to do it wrong and have people get angry at us. My long-term goal, so I am 44 now, so by the time I'm in my 60s, probably this will have happened. I'm hoping that we can get it set up here in Massachusetts, and that then we will demonstrate that it works so well, and then we'll set up this system all around the country, just like we do units right now. But I just graduated some months ago, so let's see.
So Paula asked, she gave us some information, then she asked questions. So her background, she said, I'm a dog breeder and I'm interested in how improving environment, things like better diet, less exposure to toxins, etc., would affect future generations. So she wanted to ask, how would improving diet, decreasing exposure to toxins and insecticides and heavy metals affect longevity or vitality of future generations? Can the vitality that we see in parents who are raised this way pass on to future generations? And so, again, the question is about how long, how many generations?
Yeah. It is an important and interesting question. Again, I just have focused more on behavioral aspects and less on sort of metabolism and toxin aspects. And so I don't, if there had been one study, because I knew I was going to get this question, if there had been one study that I could have read to answer it for you guys, I totally would have. Unfortunately, it's sort of this whole separate area of study from behavior, and because I focused on behavior so much, I don't know that area, and so I don't know what the classic studies are that similar to like the classic meaty study for rats that I talked about here. That's the classic behavior study, and I understand that very well, and I know that that's one of those I should be talking about. And for nutrition, metabolism, toxins, I know there's a lot of that work going on in humans, super relevant to dogs, and I just am not familiar with that literature. And unfortunately, there was no way. I mean, it would just be a case of having to read 20 different studies in order to get a grasp of it, and there was just no way I was able to do that. And I knew I was going to get this question, and I was sad because I knew I was going to have to give this answer. I just really don't know. I'm sure there's an effect. No one knows what it is. I know that there are some people who are sort of saying like, oh, giving vaccines, for example, could affect multiple generations. I would still encourage you to go ahead and give your dogs a reasonable, not too many number of vaccines, but I just can't give you a solid answer, unfortunately, about sort of what all is going on with all of that.
Sheila asked if it would be beneficial to study the brains of stillborn puppies to see if their brains show a lack of methylation, I think is what she meant, before any interaction with the mom.
That's interesting. Maybe. I would want, the thing about it is that I really want to control. Like, I'd want to be able to compare them to not stillborn puppies. I might be able to get my newborn puppies. I'm not sure exactly how I would turn that into a project that I could get. Because I've also thought about, there's this Epigenetic dog breeding clinic that we collaborate with that I'm sure would let me have things like they do C-sections fairly frequently. And so I feel like I could probably get samples of placenta, which is actually a super interesting organ for telling you about what information the puppy is receiving in utero. And I'm like, but they're going to give it to me, though, at the end of the pregnancy, and all the interesting stuff is happening really earlier on, and they can't get it to me then. And I think with the puppies, like, What I kind of want to see is whether stuff is changing in utero. And it's just harder. It's hard to get that. It's just, I mean, miscarriages. There's so few of them. So few of them happening in an individual clinic where I can go to collect info. So, anyways, I spend a lot of my free time thinking about these kinds of questions.
I'll take them out. You want to go play outside? Can you give me
CC
5
asks,
minutes?
epigenetic
5 minutes?
effects ever become genetic if the trait continues to be useful for survival?
Not directly. So there is no known mechanism whereby epigenetics can cause a mutation. I think it is certainly very likely that if a particular trait is useful for survival in an ongoing way, that then if there are any mutations to increase that trait, the expression of that trait, that would be selected for. And so you would certainly see that looking at a population over time, that if there is selection pressure for a particular trait, you're going to start seeing more and more of that trait. And you may also see that the epigenetics are supporting that as well for the same reason. But there's not a direct, the epigenetics aren't directly pushing that. It's just that there's pressures coming.
Gotcha. Brianna, just heads up, I had a question about your question, so I put it down in the comments before we get to it. We're about 2 questions away from it, so I was hoping you'd clarify before we ask it so that then we can just answer it. Paula had asked, can feeding, and this may be another one where your granddaughter know, but she says, can feeding MSM methylate epigenetics? In other words, can dietary methyls affect epigenetics in a positive or
Because
negative?
they've been giving MSM to
Yeah, I think
mice
we answered that where I basically said, I suspect there's subtle changes, not as dramatic as in the mouse study that we talked about, but I don't know specifically about MSM. In fact, I do know that we're liable to give it to dogs.
Hold on, I'm just pasting another question so I don't miss it. All right, so Sharon said that her husband and her watched 3 of Robert Sapolsky, is that how you pronounce his last name on the podcast?
Sapolsky, yes.
Many YouTube videos after listening to the podcast. They said they very enjoyed the podcast and the webinar, and thanks for shedding some light on the subject. Her question is, she's wondering if the mother dog was not fed a very good diet, if that could affect puppies and also the next generation.
Oh, sure, I would think so. And so we saw that specifically. I don't know if you were here at the beginning of this webinar, but where we talked about the Dutch hunger winter. So that was a really dramatic situation where the parents, the mother had terrible nutrition, and the human offspring showed really dramatic differences. So for sure, I think having poor nutrition can affect. And so the level of poor nutrition that you'd expect a dog, particularly a pet dog, to have, you'd see much more subtle changes than what we saw with the Dutch hunger winter, where these people were just dying at lunch. But for sure, I think the level of nutrition, good or bad, will affect the puppy.
Brianna asked, when you're talking about maternal versus paternal contributions, if there's anything studied on mitochondrial DNA contributions that affects the puppies.
Sorry.
I'm going to paste it.
Paternal. Oh, yeah. I have not seen anything about that. So mitochondrial DNA, just for those who don't know, I assume Brianna knows this. So mitochondrial DNA is mitochondria are these little organisms that millions and millions and millions and millions of years ago came to live inside of our cells, and they help power our cells and provide the energy of our cells, and they have their own DNA, and it's bacterial DNA, so it's different from our DNA in some very important ways. And so egg cells contain a bunch of mitochondria, and that is mainly what's passed on to the offspring. Generally, what has been understood until quite recently is that the sperm cell, being so small, doesn't contain any mitochondria, and hence any mitochondria is the offspring at all, and therefore when you look at mitochondrial DNA, it only shows maternal inheritance and not paternal. Although I read a really interesting series of studies a couple of years ago for a journal club turns out that sperm do sometimes have mitochondria, and they do sometimes contribute the mitochondria to the offspring, just that it's so many fewer. So as the cells divide, there's just very few. But you may actually have some mitochondria from your father, just very few. And so when we do genetic studies, we don't see it. So your question, though, I think, is about whether the mitochondrial DNA that your mom gives you is in relation to maternal effects. And I think that just falls under the rubric of straight old genetics. If that answers
So Brianna also said she would love the link to the study you just mentioned.
Oh yeah, remind me. I'll try to find that. I read that a couple years ago. You know what? You could go Google like maternal mitochondria or sperm mitochondria. You'll probably find it. But let me know if you on Facebook would be a good way. In the thread that I'll start, help me remember. I'm happy to try to help you find it, but I'm not going to promise that that'll actually be the thing.
Fair enough. Linda said, if a fearful dog takes Prozac and makes the dog less fearful, does that impact the ethics, at least in that dog, even if not in future generations?
So we don't know. We haven't studied it. We don't really know how Prozac works. My suspicion is that if a dog starts being more confident, that they're going to perceive their environment different. And therefore, their brain is going to start making changes to adapt to this less scary environment. And so I would expect that there would be some epigenetic changes, but I don't believe that's ever been looked at. So that's really just me guessing.
Fair enough. Annie asked if large litter size has effects and also androgenization of female pups in utero. I will paste that.
Yeah, androgenization.
There you go.
Androgenization. Yeah. Affects like on the offspring. So yeah. Again, I suspect that's not. Well, yeah. So large litter size is just going to provide a very different environment for the puppy when it's growing up. I think we all know that it's really tough for a singleton to learn appropriate social behavior with other dogs. It doesn't have other puppies that he's interacting with, fighting for nipples and things like that. I expect that there's probably some sort of roughly optimal litter size, and once you get a massive litter, it's probably harder for puppies, right? They're not getting as many resources from mom. There's more likely to be a bully in the litter that stresses them out. So I imagine that litter size just socially has some effect. In utero, I think you could imagine how litter size might have some effect. So androgenization, what she's talking about are these really interesting studies that have been done both in humans and in rodents, which show that in those two species, if a female, so easiest to describe in rodents, if a female has uterus with a male on either side of her, then she is born what we call androgenized, meaning she's a bit more masculine. So her genetics aren't changed, but she's developed to be a bit more masculine, and that affects things like her play style, so her behavior.
Isn't that interesting?
There's physical traits that are different. In humans, there's this weird thing where if you're androgenized, it's your fourth finger. I don't know if anyone remembers this. Like one of your fingers is longer than it would be otherwise. It's super weird. But it definitely affects, is thought to affect your behavior. And then really interesting, it also affects what the male versus female ratio of that rat's offspring is likely to be, which is very weird. So androgenization, very interesting. So far as we know, doesn't happen in dogs. And the reason is that, so humans and rats both have the same kind of placenta, in which the fetus is in very, very close contact with the mother's blood. Just one cell layer between the placenta and the mother's blood, which means that if there is a male rodent next to you, or if you are human and have a brother, that as they are starting to make testosterone and other androgens, those hormones can easily pass out into the mother's blood and travel down a few inches and then back in and get to you. And so as you're developing, you are being exposed to your brother's hormones because he's a mom and mom pass them up to you. Dogs have a different kind of, dogs and cats and carnivores in general have a different kind of placenta than we do. which is part of why birth is easier for them. We're more likely to bleed, to hemorrhage in birth because when that placenta comes away, it really rips because it's really close to the mother's blood. They just have a different evolutionary approach to it, and so they have a thicker cell layer between the placenta and the mother's blood, which means that it's harder for the mother to pass them important information. So, and I want to explain what some of the evolutionary consequences of that are, but what it actually means for indoctrinization is that because those hormones don't go back and forth as easily, it is not really possible for the male puppy's hormones to pass into the mother's blood and then to pass back. Now you may think that's strange because I keep talking about how the mother's stress hormones over. The placenta does that actively, actually. It's not passive diffusion through the blood. The placenta actually has enzymes that it makes that go and they look and they see how much cortisol there is and they push it through and they show it to the baby. The placenta is actively making decisions, actually, about, I want to let this much cortisol through. Okay, that's enough. I'm not going to let any more through. Okay, now I'm overwhelmed and more is going to come through than I really have to deal with. So in terms of cortisol, it does get actively passed through, but so far as we know, those sex hormones that are going to influence the way the level of androgenization in their treatment doesn't seem to get passed through. And so then the other interesting part about this, so I think one of the reasons why humans why humans developed, humans and rats developed to let stuff pass through so easily, is that that's how we pass our antibodies on to our children. One of the most important things that we pass on to our children the information about the pathogens that are out there in the environment, flu, cholera, whatever you're likely to see in your environment, you warn your child about it by passing your antibodies directly to you in the blood, and the child is going to see those antibodies and use them until its own immune system has developed to the point where it can defend itself from those pathogens. And dogs don't do that, which is, or don't do that as much, which is why it's more important for them to have that first milk colostrum, because the way their parents pass that information on is much more through the milk. And by the way, ruminants and horses do it even less. There's almost no contact between the mother and the baby. So that fairly quickly when I was in vet school, I would see these foals come in, and Prius, baby llamas and baby alpacas would come in, and horse foals would come in that they had failed to suckle in the first 3 days, and they had to come in, and you had to be careful to give them all the antibiotics that they need because they would have died because they didn't have their mother's blood. Okay, super long-winded answer, which gave you a lot more than you asked for, but I really like people to know that about androgen. Is a very common misconception in the dog world that just because it's true in humans and rats that it must be true in dogs. Actually, a reason why we wouldn't expect to see it in dogs, and that's because of that physical difference in the placenta.
Interesting. It's really
and thank you, Linda, for saying that. Linda just had a comment about saying thanks for sticking around. I actually like answering the questions. It's one of my favorite parts.
It's like
We've got a couple more here for you, so we'll see if
I'm done. Stop. Okay.
Paula asked to follow up on the length of generational changes question earlier. She said somewhere she read that studies were done on hundreds of generations in insects, like tiny flies, and the changes were affected many generations.
Oh.
She was curious if that was true.
Oh, I have not heard of those. That's really interesting. Never heard of them. Possibly. That would be a good model organism to do, I think, because their generational time is so short that it would be easy to follow them hundreds of generations. I mean, again, as I said, I would be a bit surprised to see information passed along if the environment was different, because it just doesn't seem to me to make sense that that one animal would have an experience. It makes sense to pass it on to your offspring because your offspring are likely to be in the same environment, but if that offspring then discover that they're actually in a different environment, it doesn't make any sense to me that they would pass that information on about grandpa's environment. The environment has changed, which it is very likely to do. So I'd be a little surprised, but maybe I haven't seen those studies.
Interesting. It'd be interesting in flies since they have such a short lifetime. It was different because maybe it doesn't make more sense for them.
Yeah, maybe it's right. Maybe it's different, right? I don't know. It's interesting, and there's just so much we don't know as well. So all the things I'm telling you could be wrong. It's just
So Paula asked, she said she always sees a graphic with the spooling and unspooling. And wanted to know, I don't know if you know what graphic she's talking about. She said, could you please explain at a lay level whether the spooled up genes are affected negatively or positively? She doesn't understand what is positive versus what is negative. In other words, is the epi gene triggering a good thing or a bad thing?
Let me see if I have, so I have this even more detailed graphic, which I saved because I started to put it into the talk, and then I was like, oh no, but I think this actually explains it better, and I just thought it was too much. But then I saved it just in case someone asked this exact question. All right, so here you have the DNA, and you have it spooling around the proteins. So remember my example of the airplane that wants to try to land on DNA. So the airplane comes down here. So here's where they put, right? The airplane tries to land here and express the gene here. And so here they have this note. The DNA is accessible and therefore the gene is turned off, right? The airplane can get at it, RNA polymerase can get at it and can express the gene. Here it's all spooled up and they have a note, DNA inaccessible, gene turned off. Why that font is so much smaller, I couldn't tell you, but I hope this answers your question.
I will see if she follows up. I'm not sure if she's still with us or not. But Paula, if that didn't answer your question, let us know, and I will see if Jess. Oh, she's still typing, so she must be with us. We're gonna have a second, just in case she's got to follow up there. Showing me her having stopped typing. I'm not sure if she hit enter and it just hasn't shown up for me yet, or if she's stopped.
She stopped, and then she heard you. So there, keep going. Yeah, so people are, while we're waiting for her, people are talking about the possibility that flies have this lack of, she doesn't understand if it's negative or positive. Yeah. Mm-hmm. So DNA accessible gene turned on, so that's positive in the sense that more of the protein will be made. DNA inaccessible gene turned off, that's negative in the sense that less of the protein will be made. Does that answer your question?
Yes.
Perfect. Good.
So you were going to say something about what folks were commenting on.
Oh yeah, so people are talking right now about the possibility that the fly study, that it may be that since flies don't mother, there could be differences there. The stuff I've been talking about has been in like humans and rats and mice, and we all mother. Yeah, I'm trying to think if I've read any epigenetic studies done in species that don't mother. I think I have. So that's possible. I don't know.
Interesting. All right, I got you back. Pitch first from me, perhaps up there. So Chrissy asked, are susceptibility to addictions like alcoholism in humans and maybe ball junkies in dogs genetic or epigenetic?
Yeah, there's a massive literature out there on addiction. And by the way, the person that I had lunch with today, whereas she's collecting the dog brains for us, she's really interested in addiction. There's definitely genetic predilections to addiction in humans. I have no idea what the epigenetics of addiction might be. I haven't done any reading about that. I've got to think that it's been studied because it's very well, they have really good mouse and rat models for addiction. So it would be pretty easy to study, to do the kinds of invasive, like looking in the brain studies. So I've got to think they must have done it, but I haven't seen those studies. I would think they'd be out there. Yeah. Don't know. You guys do ask good questions. It makes me really sad every time when I can't answer them.
So Anne says she's learning about the puppy culture or early neurological stimulation being beneficial to puppy resiliency. And she wondered if some sort of neurological stimulation for a pregnant female would also be beneficial in terms of stresses that are not all negative.
That's interesting. I haven't seen any studies that have looked at that one way or the other. I think that at one level, certainly keeping the female enriched and happy. So basically what you want is you want the female's environment to be as similar as possible to the environment that the puppy is going to experience. So obviously you're going to want the puppy to be in an environment that's safe and happy, but not completely boring, right? And so if you have this female and you're like, oh, you're pregnant now, you can't get stressed out at all, I'm just going to quit you for the next 2 months. That's not so good either, even if she were happy being created, which probably that would be pretty stressful for most dogs. But even if that were the case, she'd then be passing on information. Theoretically, I don't know exactly how this would work, but theoretically, she'd be passing it to the puppy, saying, "Okay, well, apparently the world that you're going to experience is super quiet, and there's never any loud noises, and there's no other strange dogs." So really, what you want is sort of like when you're socializing a puppy, right? You want the pregnant female theoretically to experience sort of an ideal world that she can pass on to the puppy. And so there's going to be some mild stressors in that, and that's okay for her to pass it on to the puppy. It just can't be something that's too much. I'm a little worried that you guys are going to take that and turn that into specific advice on how to manage a pregnant female. And I just wanted to make the point that I don't. First of all, I don't believe there's been any science on how to manage pregnant female dogs. I think that'd be really great science to do. It's not been done. So I'm not sure that anybody knows, and I definitely haven't really done specific thinking about that. So just as generalities.
So the last few are pretty quick questions. I only have 2 more questions though, guys. So if anybody does have a question, they're really, really hoping to sneak in there before we're done. Although I'm sure Jessica would love to go to bed in the next few minutes. I'm going to ask these last 2, and then if anything else comes in. Rosie wanted to know just if you remembered when the paternal mitochondrial DNA paper came out. She remembers the 2012 paper about paternal mitochondrial DNA degradation in early embryo development.
That may be the same paper. So the paper that 2012 is very likely because the journal club that I read this paper for would have been like 2013, 2014, 2015, something like that. And we only read really recent papers for it. They were really obsessive about that. So 2012 is very likely. And so if we're thinking about the same paper, what it was so cool. They attached fluorescent markers to the mitochondria, and so they had these pictures of the embryo dividing. And so first it's one cell, and you can see the fluorescence of the dad's mitochondria being in there, and then it's like by the time that it's 4 cells, and it's like, oh, okay, in this cell it didn't get any of dad's, but here's some of them in these other cells, and then you could see by the time I don't know how 32 cells or something, like, oh, okay, and now by this point they just could hardly see it at all. So it's super cool, like just seeing it get sort of diffused out, diluted out. But so maybe that's the same paper. I don't know if that's enough information, but yeah, probably 2012.
All right, so last one, and this is a good one to end on. So Emily had asked, will the June course be appropriate for totally non-sciencey types who think it's all interesting but can't repeat any of it back even under threat of death, asking for a prize?
I mean, I guess you have to ask if this webinar was appropriate for people like that. So I make a real effort to cater to everybody. So I try really hard to cater to people who don't have any sort of biology or genetics background. So I try really hard to always provide metaphors like the airplane and the bookmarks and stuff like that. But then I also try to cater to people who have a biology or genetics background and want to go in deeper. And so I try to probably irritating everybody, right? So I try to start out with giving a solid background and then sort of dive deep in at a couple of points, come back, and then dive deep in again, and then give a good summary at the end. And so hopefully everybody can tolerate the bits that are not aimed at them, but hopefully there's something for everyone. And I absolutely will do the course the same way. So the way we're going to structure it, I think, is we're only going to have bronze and silver, no gold. And the way silver will work, I'm thinking, is that basically each week the homework will be sort of trying to apply what we've learned to your own dog, and it will be absolutely if you have no biology or genetics background, I'm more than happy to work with you on that. That would be a great place, though, to make sure that you get your questions answered exactly at the level that you want, because once I know who I'm talking to, I hope I'm very good at adjusting to give exactly the information that they want. Hopefully I'll be able to, for the bronze students, obviously I won't get any feedback on, I'll do my absolute best to cover all the options, but it's obviously really hard when you're not getting feedback from your audience to know if you're doing a good job or something. I'll say this though, I'll say this is that if somebody does do the Facebook group, I'll try to poke my nose in there, not answer questions, but just to make sure that I'm on track. And so if there were a Facebook group associated with it for the bronzes, and people were to say there something like, oh my god, I can't pull this off. I would try to poke my head in and be like, oh, I better do a better job.
Carly commented that she had chemistry and such in college, but only high school bio, and she could mostly follow. So she might have to read or listen twice, but she could mostly follow. So, and Rosie said you're very good at talking to the level of the person.
Oh, thank you. Thank you. Proud.
All right, so we are through our questions, guys. I hope we'll see some of you guys back next week for Mariah's webinar on reward placement strategies. Get your cookies here. And with that, we are going to call it a night.